how can we more effectively treat cancer? Cancer is normally treated using radiotherapy, chemotherapy, or some combination of the two. Radiotherapy is the use of ionizing radiation, usually X-rays, to kill malignant cells. Radiation is used with the aim of damaging the DNA of cells, ultimately leading to cell death. To minimize damage to surrounding tissue, radiation is often applied in the form of several overlapping beams focused on the tumour. Fractionated doses can also be used, which allow healthy tissue some time to recover before the next radiation dose is applied. Chemotherapy involves the use of drugs that kill rapidly dividing cells to damage cancerous growth. Newer chemical agents target specific proteins in cancer cells to minimize damage to normal rapidly dividing tissue. Unfortunately, these methods don't work in all cases. Some cancers are radioresistant, some are immune to chemotherapy agents, some are too close to sensitive organs to be accurately targeted. The list goes on. So how do we combat these complexities? Through better targeting, more effective drug delivery, or dose enhancement specifically in the cancer? My research is focused on enhancing the effects of radiotherapy. The current aim in this field is to dramatically improve the potency of radiation in the cancer cells while leaving healthy tissue unaffected. This is a nanoparticle. The figure shown is under significant magnification. It would take 3,000 of the nanoparticles I regularly use to encircle a single human hair. A nanoparticle is defined as a small object that behaves as a whole unit with respect to its transport and properties. The nanoparticles we use are small clusters of atoms of a heavy element. Heavier elements absorb X-rays more readily than lighter elements, such as those that make up the human body. When X-rays are absorbed, these nanoparticles give off electrons, which can lead to DNA damage. Nanoparticles have irregular surfaces, to which we can attach different chemicals, such as sugars, chemotherapy agents, or enzymes that penetrate cell membranes. If we can send these nanoparticles specifically to cancer cells, we can drastically improve the effective local radiotherapy dose and divert chemotherapy agents away from healthy tissue, reducing any collateral damage during cancer treatment. My research has two sides to it, an experimental side and a modelling side. On the experimental front, I have helped to build new experimental apparatus which allows the throughput of several hundred times more samples than our group could previously irradiate and analyse, which has increased our data collection by a large margin. We have shown that the presence of nanoparticles significantly increases the generation of DNA-damaging species and seek to improve this further. To this end, I have recently produced a model which describes the currently accepted pathway for enhanced DNA damage in the presence of nanoparticles, but this model shows significantly less enhancement than we observe experimentally. Something else is happening here. In light of this, we suggest the existence of a new DNA damage pathway in the presence of nanoparticles that isn't just to do with electron interactions. Our recent results show that this new pathway is even more important than we first thought. Here, we highlight the change in the ratio of contributions from this new pathway, C, and the traditionally accepted pathway A, as the accuracy of my model improves. Thankfully, the combination of advanced computational modelling with our enhanced experimental throughput and precision gives us the unique ability to catalogue the contributions of many nanoparticle materials and further determine the origin and nature of this new pathway. This new information is a major part of a large, multidisciplinary collaboration that directly informs radiographers and oncologists, leading to more effective cancer treatment.